Today we're going to be covering TMV, polio, and yellow fever. So the three virus families that we've gone over. Give you a brief overview or touch on some of the more challenging concepts of the three families that will be on the midterm. So hopefully that's helpful for everyone. So again, we're going to look at our friend here, the tobacco mosaic virus. So a brief overview here. It has a helical capsid structure. It has a single-stranded RNA genome, just like every virus we're looking at today. The genome structure, again, is linear, and it isn't enveloped, so it's a naked virus. And the three strategies that we're going to take a deeper look at today is going to be its movement proteins, which is very important for the midterm and testing, the leaky stop codons in its genome, and as well its subgenomic RNAs. So here's a brief overview of the tobacco mosaic virus genome. So we're going to take a closer look at the different strategies that this genome uses, this virus uses. Tobacco mosaic virus, it has what we call a leaky stop codon in its first open reading frame and second open reading frame. So what this means is when the RNA is directly translated into a protein in the cytoplasm, sometimes a stop codon is missed by the ribosome, so it can continue on translating past the ribosome. Now, most of the time this doesn't happen. So most of the time we have it where we have translation from the 5' caps through the MTR to the helicase. But about 17% of the time, we actually have it where it continues on all the way to the end, and we get this full length 183 kilodalton protein that includes the RDRP on the end. So that's going to be the polymerase domain that the virus is going to be using in order to make more RNA, whether it be for mRNA or to replicate its genome. So the resulting proteins are going to be the 126 kilodalton protein, the MTR and helicase, and then the 183 kilodalton protein, so the MTR, the helicase, and the polymerase RDRP. So what happens is with the percentages that this leaky stop codon works and doesn't work, we end up having one of these full length proteins for every 5 of the shorter ones. And then these actually form this hexameric ring. So where the helicase domains are, they all line up and they're actually associated with each other, forming that ring. Now you might say, well, wouldn't it be smarter for the virus just to make this full length protein, right? So you would have 6 times as many polymerases. But the difference is that when we actually have this hexameric ring structure, it increases the polymerase activity tenfold. So we actually have 10 times more activity when it's in this hexameric structure, and 10 times more RNA can be produced when this system is in place. The leaky stop codon is the first way that the tobacco mosaic, or the first viral strategy that it uses. The second one is going to be something called subgenomic RNAs. So what happens is when we have that polymerase, it will take the positive sense RNA genome, and it will replicate that into a negative sense. And then from there, we're going to have these subgenomic RNAs, which become messenger RNAs more or less, created from that negative sense RNA genome. And the creation of these subgenomic RNAs starts at these 2 different subgenomic promoters. So what happens is the RDRP will hop onto the negative sense at these promoters and create these 2 messenger RNAs from ORF3 and ORF4. So we have this full length one, and all this does is it allows the translation of the movement protein, and then this smaller one is translated into this capsid protein. All right, so movement proteins are one of the vital and important discoveries of TMV. So movement proteins are something that most plant viruses have. And I really urge everyone not only to read the original paper that discovered movement proteins, but also the updated one, just because there's going to be so many questions about that in the midterm, in quizzes, exams, everything like that. So you really should have a good working knowledge of that. But the overall model that we understand of tobacco mosaic virus is we start with the actual viral entry. So with a lot of plant viruses, It's usually a mechanical transmission. So if the plant cells are damaged, it allows the virus to get in, or a virus or a vector like an insect is able to break a cell, and they also have the virus carried with them. So there's plenty of ways for the virus to get in. After that, you're going to go through viral uncoding and protein expression. So all the capsid proteins are going to break off from the genome, allowing it to be replicated and also to be used to create its various proteins. So the RDRP, the CP, and the movement protein that we discussed. The next step is the viral RNA, and the proteins will actually localize to the ER. And at this point, this is the old system now, we actually would see the movement proteins and the RNA kind of complex together, and they make this thread-like structure, okay? So from there, this thread is actually going to be trafficked on actin strands, so the microtubule network of the cell through these myosin motor proteins, and they're going to be trafficked toward these things called plasmodesmatas. Now, typically they're pretty small tubules or connecting structures between two plant cells, and they have this p38 protein in them. So what's going to happen is this thread structure is going to be brought to them, and the movement proteins of the structure is going to interact with p38, and it's going to allow the size exclusion limit of this tubule to expand. So it'll grow larger, which will allow the RNA and the movement proteins to go through. At this point, though, the movement proteins in the old model would be attached to p38, and they would actually need to be phosphorylated by an intercellular kinase to be released, and that actually allows everything to be released into this adjacent cell. And then from there, the RNA genome can undergo translation and replication and carry on its infection cycle from cell to cell to cell. Okay, so this is the old model, and then we look at the new model here. So the new model is similar, but the only difference here is researchers looked at where in the cell all these different proteins were localizing, and they actually found that they were also localizing to plasmodesmatas as well. And that kind of helped us understand that it wasn't just the RNA and the movement proteins that were localizing there, but it was actually these entire viral replication complexes that actually took off sections of the ER membrane to form almost like factories for new virus. So the same thing here. So the protein and the RNA for the virus would localize to the ER. It would create this complex, and this complex would actually be localized or trafficked towards the plasmodesmata through myosin. And then from there, it would interact with the plasmodesmata and allow the entire complex to squeeze through into the adjacent cell. So we have a viral replication complex, which then ultimately becomes this viral movement complex and is able to infect this adjacent cell. So it's a really neat strategy, and it's almost a different form of virion for this virus and how it can move from cell to cell. So I really want to emphasize that you should know both the papers that are related to tobacco mosaic virus that Dr. Mung mentioned in lecture, and have a really good understanding of how they compare and contrast and how they've developed, and how our idea in science can change. Okay, so polio virus is going to be our example virus for Picorna viridae. So different than tobacco mosaic virus in the sense that it's an icosahedral viral structure. Its genome is still positive sense RNA, still linear, and it is still a naked virus. It has a number of interesting viral strategies. So it has an internal ribosomal entry site. It has a viral protein genome linked, so a VPG. It has a polyprotein processing system, and it also is able to silence the host translation. So let's take a deeper look at how that works. Or what this the polio virus does is it has a single open reading frame, which is translated into a large protein, and then from there, a number of proteases, so viral proteases, and possibly a cellular protease work to basically to cleave up this protein into smaller functional proteins. So if we look at the capsid structure here, or the capsid section of the genome, we have VP0, which is cleaved into VP2 and VP4. VP3, and VP1. So that goes back to actually the components that the virus needs in order to make its capsid and the different facets. And then if we look at P2 and P3, it gets cleaved into all these other proteins. So 2A pro, which is one of the proteases it uses, 2B, 2C, and then also this protein 3, or this section here, is cleaved into 3AB, 3CD pro, and then 3C pro, which is another protease, and then 3-pol, which is a polymerase of the virus, and then 3A and 3B are part of the VPG structure, or the viral protein genome link that we're going to be taking a look at. But basically what this does is it allows the virus to pack a lot of genes into a relatively small genome. So I'm just going to cover replicative form and replicative intermediate just quickly, just because I feel like a lot of people kind of got confused by this in the lecture itself. So the replicative form is a double-stranded RNA byproduct of RNA replication. And that's actually generated by the polymerase that transcribes a complementary negative single-stranded RNA from the positive sense RNA genome. So if we look at that here in the diagram, so we have this positive sense RNA strand, and then with RNA-dependent RNA polymerase, it's able to create that negative sense single strand on that to create that double-stranded DNA, or the replicative form. We also have the replicative intermediate, which is when the RF itself is unwound to separate the single-stranded RNA from the positive sense single-stranded RNA. From there, the negative sense RNA is actually used as a template, and we get these multiple positive sense RNAs transcribed from it, or replicating the genome for the virus. So we can see that here we have all these different polymerases on the replicative intermediate that's creating these additional pieces of positive sense RNA. So I'm just going to go through a quick review of how our host expression works, just so we can kind of see how the virus then attacks that and shuts that system down. So there's a few things that are super necessary to host expression and host translation. So we typically start this first step with this the pre-initiation complex at the 5' cap here. And we have this cap binding protein, this EIF4E, and that will bind to EIF4G, which is almost like a scaffold for the rest of these initiation complex proteins. And as well, you can see this 40S subunit here, which is the smaller part of the ribosome, is also in this complex. So what happens is ATP is going to be used up to make ADP and inorganic phosphate, which will actually give this complex the energy to start moving down the piece of positive sense RNA. And it's going to be scanning for that start codon, AUG. And that start codon is going to match up with this, what's the word I'm looking for? tRNA? tRNA, right? Yeah. Geez, okay, sorry about that. With methionine, and that will actually be the start of translation, right, at that stop codon. And then from there, once it finds that section on the RNA, that 60S subunit is going to come along. The rest of these initiation complex proteins are going to be removed, and it will allow the ribosome to start actively translating that RNA into a protein. Okay, so I'm going to break everyone out into their breakout rooms.
A few minutes later.
The cell is infected with poliovirus, so that's really key to answering all of these questions. So based on your understanding of the actual impacts of poliovirus on the host cell that you learned in lecture, answer the following questions. So the first one here is, can a single-stranded RNA with a 5' cap be translated by an infected host cell ribosomes? So the answer to that one is going to be no. And the reason for that is the single-stranded RNA, positive sense, with a 5' cap is basically imitating what a host mRNA would look like. And because that host cell is infected with polio, that conventional translation system will actually be silenced. So that's why that wouldn't work. The next question here is, can a negative sense replicative intermediate be directly translated by the infected host cell ribosomes? So the answer to that one is also no. And the reason for that is because mRNA that gets translated into protein is made of positive sense RNA, not negative sense. So we wouldn't see that occur. The next question is, can a single-stranded positive sense RNA with an IRES be translated by an infected host cell ribosome? And the answer to that one is yes. And we'll get into a little bit more of an explanation of what an IRES, so that's an internal ribosome entry site, is. But basically, it's a way for the virus to get around the host expression system that it's actually shut down. And then the last one is, can a double-stranded RNA replicative form be directly translated by the infected host cell ribosomes? And the reason for that is a little bit different, but basically the answer is no, but it's because it's actually energetically unfavorable to unwind double-stranded RNA simply just to translate from it and then rewind it back. So what would happen is you would actually see it, that double-stranded RNA break apart for the negative sense piece to be transcribed into more positive sense RNAs, and then the RNA, the positive sense RNA would be used in order to actually translate proteins directly from that. So if we go to the next slide, we're going to break down all these different ways that the polio virus shuts down that host system. So picornaviruses block transcription and translation of the host RNA. So the first way it does that is it uses that TA protease that we discussed before, and it actually cleaves the eukaryotic initiation factor 4G. So that's the scaffold protein that you need to start cap-dependent translation. So we see here EIF4E is bound to this EIF4G scaffold protein, and then EIF4A is here. And then 2A comes along and just chops it right off here. So it can't actually form that cap initiation complex. So the next way it actually shuts down translation here is poliovirus infected cells have a dephosphorylated 4E-BP1. So that's basically it's an EIF4E sequestering protein. So this cap binding protein here, when this protein is dephosphorylated, gets bound to the 4E-BP1, and then we're no longer able to create that the initiation complex because we can't bind to the actual cap of the 5' cap of the positive sense RNA. And also a number of other viruses, picornaviruses, do this. I believe what is it? HCMV, I believe, was the
encephalomyocarditis.
There it is. That was another example given in lecture. So just be aware, we don't actually know what the actual mechanism is. It might actually be a host response from being infected, but this is ultimately not a big deal for the polio virus because it has the IRES, and we're going to get into what that does for it exactly. So the next way that it actually silences the host expression is that 3C or the 3C protease cleaves the TATA binding protein. So everyone should remember that. Oh, thanks for that, Catherine. So what that would do is when that protein is cleaved and inactivated, it would actually block host transcription. So typically in DNA, we'll find this TATA box, the TBP, binds to it, and then the eukaryotic transcription complex forms around that. But when that's cleaved, we don't actually see that transcription of mRNAs from the host cell genome. So now we're finally getting into what the IRES is. So we're going to look at the Pelletier and Sonnenberg experiment. So what they did is they took this strand of RNA here, and it has 2 open reading frames. So they found the majority of the proteins created were from open reading frame 1, and a very, very small portion was from open reading frame 2, which is pretty normal for eukaryotic cells because most mRNA is monocistronic. So we kind of covered that briefly in lecture today. So really ribosomes only have the capacity to translate 1 open reading frame, and then they hit the stop codon, and then they typically fall off. But every now and then they actually have the ability to read through that, and that's why you would see that small amount of protein from open reading frame 2. But they took the 5' untranslated region of poliovirus, so 742 bases long, and they actually inserted that in this piece of RNA before open reading frame 2. And what they found is the protein that was then translated off this piece of RNA actually had a lot of protein from open reading frame 2 as well as open reading frame 1. So they were kind of confused by that. They didn't know what that meant exactly. So they then followed the experiment up with the next stage, is where they did the same thing with that piece of RNA with and without that IRES inserted. And they found that only protein from open reading frame 2 was translated. So what this means is it's a way that this protein can still be translated and completely doesn't at all rely on that cap initiation complex. So we can kind of see a representation here, the conventional eukaryotic 5' cap. In the polio infected cells, nothing is translated, but then with the IRES, we do actually see that creation of protein here. So I'm going to get into a little bit more detail about the IRES itself. So as we've kind of been over here, so the polio virus genome lacks the 5' cap, and therefore it can't rely on cap-dependent translation. But it has this IRES, which is actually, it's not a conserved sequence of RNA per se, but what it does have is a conserved secondary and tertiary structure. So we get all these kind of weird knots and loops and everything, and that's almost like a net or fingers that can reach out and grab the 40S subunit of a ribosome and pull that onto the start codon of the open reading frame. And we also see another common feature is these pyrimidine-rich regions. And as a result, we can somewhat use those different structural elements to classify these IRESs here. So as we can see here, the 40S subunit is pulled by the IRES onto the open reading frame, and then from there, the 60S subunit can join on and translation can begin. And then here as well, back to the point where we don't actually see a lot of sequence conservation, but we do have secondary structure conserved. And these are the 5 main types of IRESs that we see here. So the next thing we're going to be looking at is VPG. So this is the viral protein genome linked. So what this ultimately allows is it allows the poliovirus genome to be replicated, and it uses that VPG to do that. So what starts is, or how it starts is this 3AB protein that comes from that big polyprotein of poliovirus. It actually localizes and it attaches to this polio, or to this ER membrane. From there, the 3B protein is post-transcriptionally modified by uridylation, and that's by the viral protein 3D polymerase. So when that happens, 2 uracils, so these 2 U groups, are added on to the hydroxyl group, which is on a tyrosine residue of the 3B portion of the protein. And then from there, these uracils anneal to the poly A tail, right? So the complementary poly A tail of the viral genome. And then 3C protease comes along and actually cleaves that 3B section from the 3A protein, and that's how we get that VPG. So it's a viral protein that's linked to the genome, and that actually allows the polymerase to come along and then continue to transcribe the negative sense strand of RNA. So this one's a little bit tricky to kind of see the whole process, but I hope this kind of representation and step-by-step process makes a little bit of sense. So after this, we're going to move on to flaviviridae, and this is one of the earliest human viruses that we know of. So this is the yellow fever virus is going to be our representative here. So the viral strategies it uses is, again, it uses the polyprotein that it cleaves to make functional proteins. It also couples translation, VRC formation, and assembly at the ER membrane. So it keeps everything in a tight little space, so everything is done very efficiently. And then it also has a really interesting protein called the E dimer, and it changes throughout the course of the replication cycle to accomplish many different things for the virus. So we're going to go over that. So here's the genome here. So again, one big open reading frame, and then that's translated into this larger protein, and then using either host cell proteases or viral encoded proteases, it's able to chop that up in different sections to make functional proteins. So if we look at this one here, so this is the hepatitis C virus, yellow fever virus, and other members of the Flaviviridae or flavivirus genus share certain features. So again, a single open reading frame with the polyprotein, and then that's post-translationally cleaved by host proteases as well as viral proteases, but they do have differences among them, okay? So when we look at yellow fever, it actually does have that 5' cap again that you can see over here, but then viruses such as hepatitis C virus, they actually use the iris. So there are subtle differences between different viruses in the same genus, okay? So keep that in mind. So they're not all 100% similar. They do have very significant differences in their strategies. So this is ultimately what the big polyprotein looks like, or a representation of that. So if you remember back from MCB 2050, I guess would be a good example. When a protein is translated into the ER, it can kind of create a topology where it loops back and forth through a membrane. The virus actually uses this in a really interesting way because it loops back and forth in order to use either viral proteases or host proteases. So proteases that you would typically find in the ER lumen, it uses such as signalase and furin, and then this other ER protease that we aren't quite sure of. But what it does is it puts the proteins in a certain topology where the sections that need to be cleaved by those proteases are inside the ER, and then the proteins that can be cleaved by the viral proteases are in the cytoplasm. So everything's accessible, and it's a very controlled system in that sense. So ultimately, when everything is cleaved and assembled, these are the conformations that the virion would assume. So this would be extracellular. So we can see here it's a naked, or it's not a naked, but it has the viral capsid here, and you can kind of see the RNA underneath that, and then that's surrounded by an envelope membrane, and the membrane actually has these E dimer proteins, these M proteins, and then obviously the plasma membrane. But then we also have a different conformation, which is typically affected by a change in pH, where these E dimer proteins can break apart and do different tasks in this monomer structure versus being found in a dimer. I'm going to get into how that actually happens. So when the E dimers are still formed, the virus attaches to the host cell through that viral protein. And then from there, a clathrin coat is formed intracellularly, and that actually brings in the virus. So that's the entry step. And then as we know, the next step after that is going to be encoding. So the virion containing vessel is transported through the trans-Golgi network. And then as it does that, endosomal acidification occurs, and that actually changes and affects the conformation of this E dimer protein. And the two overlapping parts of the E dimer stand up away from each other, and that actually exposes a fusion peptide on the E dimer, and that can attach to the cellular membrane of the vesicle, and that actually allows the vesicle to kind of fuse with the endosome, and that allows the virus genome to uncoat and break out and then carry on the next stage, the replication cycle. So it's a really interesting protein. It's very, very classical among Flaviviridae. That brings us to the end of the tutorial for today. So again, I really just want to emphasize that everyone should again really look at the new and old model for TMV, be really aware of those papers, give them a read through.